Hello, my name is Shane Dunn. This is my presentation for my BIO-520 class. It's on blood. If you're one of my students, please subscribe to this video unless you need to help blood for BIO 210 or 202. Come on. So here's the functions of blood. Blood is to transport oxygen and nutrients. It is to help form blood clots, to carry cell bodies and antibodies, carry cells and antibodies, bring waste products to the kidneys and liver so that it can be eliminated, and regulate body temperature. Here is constituents of blood. So blood is composed of mostly plasma, 55% of it's plasma. Most of that plasma is water. There are some proteins, mainly albumin, which we'll talk about later on. Albumin is very, very big for maintaining blood in blood for blood pressure and such. We have electrolytes, and we have gases, so oxygen, carbon dioxide, as well as nitrogen. Then we have this kind of buffery coat. Inside the buffery coat, we have platelets, which are going to be also known as thrombocytes. And these thrombocytes are going to be the things that actually help clog blood and the sticky tack that uses blood to make blood clots. We also have leukocytes or white blood cells. Well, I'll talk about more of those next week as we do the immunology, but white blood cells are help fight infections. The second largest one is erythrocytes or red blood cells. Red blood cells' main goal is to transport oxygen to tissues and to the lungs. They also aid in forming blood clots because the platelets kind of make them force them to. So let's talk about erythrocytes, because this is the biggest part about blood. So they are biconcave, and what that basically means, oops, I might color right here. Let's use purple. Biconcave is they have this kind of dent on both sides. So they have no nucleus. So when blood cells are mature and they're formed, the nucleus is scooped out and almost all components is scooped out. So that's why their only function is to transport blood, is because they have no nucleus. They can't make proteins. So that's all they can do. Because of this, they can only survive 120 days or until they're damaged. After 120 days, most red blood cells get damaged. So they don't last for very long, but that's good because all we need to do is to use hemoglobin to carry blood. So this molecule over here is hemoglobin. Hemoglobin is a quaternary protein, which just basically means it is 4 tertiary or complex proteins stuck together. There is 2 alpha chains and there's 2 beta chains. And it's inside each of the hemoglobin molecules, which is a single chain, or globin molecules, is a heme group, and on the heme group is an iron. And that iron molecule is what actually makes oxygen attach and makes CO2 attached. Blood is made in red bone marrow, and this hormone called erythropoietin, or EPO, is the hormone that is released by the kidneys to help tell red bone marrow to make blood. Interesting enough, EPO is actually used by some athletes to actually dope, it's called blood doping, to make more blood. It's a naturally occurring hormone, but if you take too much of it, you have more blood. I'll talk about that a little bit more. When 120 days are up or when a blood vessel is damaged, when blood cells are hurt, they go to the spleen and liver to be destroyed by macrophages. Macrophages destroy things by eating them, so think of Pac-Man, kind of eating the red blood cells. So here we have hemoglobin saturation, and what this just means is that hemoglobin has a hard time getting started, but once it gets started, it goes off. So let's go back to this heme group, and the best analogy I have, it's kind of fun, but I know we're all GCU students, so we don't do this, but some fun stories when you're not a GCU student is you have 4 friends, you all go out, right? Let's say you go out drinking milk, and you call an Uber, and after being wasted on milk, one person calls the Uber, right? Well, as soon as one person gets in the Uber or one oxygen molecule attached to a heme group, right, it causes a shift in all the other heme groups, allowing oxygen to flow in much more. I know this analogy doesn't make any sense, but when we look at the graph, what I'm trying to get at is it takes a lot of energy for, where is my mouse? Sorry about that. For one oxygen to attach, but as soon as oxygen attaches, it exhalates very quickly because the conformational change will just get off. And what that means is heme either wants full oxygen or no oxygen whatsoever. There is no middle. So once you're in a oxygen deprived state, usually in the lung, once blood is in an oxygen deprived state, usually in the lungs, it takes in one oxygen, and all the shape, it all shifts, taking way more. Meaning that way it can have the oxygen and it'll hold on to the oxygen into an oxygen deprived state, usually muscles or cells, and kicks them off at once. That way the heme group is either satisfied with oxygen or has no oxygen whatsoever. The same thing is true with CO2. What this left and right thing is trying to show is once you're in an oxygen deprived state, right, you are more acidic because CO2 is a gas. So if you're in a more acidic state, you kind of shift towards the right, and that just basically lowers all energy, making it easier for you to kick off oxygen. If you're in an oxygen deprived state, all the way around. Okay. So here is blood typing. We have 4 types: type A, type B, type AB, and type O. Type A blood will have type A antigens. So antigens are the sort of thing around the cell that when white blood cells bump into, they can recognize. So all A blood types have A antigens or A faces. So when a white blood cell bumps into it, it goes, oh, that's A. If you're type A, that's supposed to be there. In the blood, you'll have anti-B or type B antibodies. These will recognize B antigens, freak out, and swarm B blood, causing a coagulation to occur. Type B blood has B faces or antigens, and it has, oops, my bad, A antibodies. Type AB antigens have, type AB has AB antigens, so it has both of them, and because it has both of them, has no antibodies whatsoever. Type O has no antigens and has both antibodies. And lastly, we have these Reese's factor, or RH factor, which is the plus or minus. So let's look at this blood compatibility test. So if you notice, if you are positive, right? So if you're type A or A positive, you can give A blood or AB blood. That's who you can give to. You can give to A positive or AB positive. You can receive A positive, A negative, O positive, O negative. What this means is if you're positive in the RH factor, you can receive negative blood as well as positive blood. If you're A negative, you can only receive negative blood, A negative blood, or B negative blood. So let's look at this. Sorry for the slide. So let's look at the universal giver and the universal receiver. So type O negative is called the universal donor because everyone can take O negative blood. And then AB positive is called the universal receiver because it receives all the other blood. So let's do this hypothetical example. I am A negative. So if I am in an emergency, right, what blood type would they give me if they don't know my blood type? Let's say I don't have a blood card on me or a wrist bracelet to my blood type. Hospitals will always have on stock O negative blood to give to patients until they can do a blood type test. Because if you give the wrong type, the blood will clot, coagulation will occur, and the person can die. This is very, very bad in pregnancy. This is RH incompatibility. This mainly occurs with an RH negative or negative blood type mom and a positive baby. Because the RH positive blood types do not have the RH antibodies, they don't react at all, but the mom has the antibodies. And her antibodies can pass through the placental wall, the umbilical cord, and actually cause the baby cells to start dying because of the antibodies. So this is why this is the main cause of miscarriages in third world countries. Luckily, we can kind of tone down this stuff with drugs. If females have this, is why prenatal care is very important for females. So it's about blood flow. So resistance and blood flow. So things that can cause increased resistance is blood viscosity. So if you have more red blood cells or thicker blood, so either more red blood cells or there's less plasma and the blood's just thicker, it's going to make the blood harder to move. If the vessel is shorter, resistance goes down because blood can flow more easy. The longer a blood vessel is, the more resistance going to be in there, because resistance is defined as blood hitting the side of a wall. So the longer a blood vessel is, the more chances blood of hitting that side of the wall causing pressure. And then radius. So right here is an example of vasodilation. So that's when blood vessels get bigger, and that will decrease blood pressure or resistance. Vasoconstriction is when blood vessels shrink and there's more room for the blood to hit the walls because blood can't flow as much. So it's actually going to cause increased blood pressure, resistance. We also could have plaque buildup, which causes the blood vessels to not go in a nice straight line and causes some turbulence to occur. This can actually increase blood pressure and blood resistance. Here's an example of pressure gradients. This is very, very big in lymphatic tissue, but basically blood flows from the arterial end to the venous end, and in between there's capillaries. So right now we are looking at a capillary, and blood comes in the arterial end, and since it's being pumped from the heart, you have all this pressure coming in. So all this hydrostatic pressure is going to push some of the blood out. And it's going to let its oxygen come out, and nutrients come out, right? But you still have this osmotic pressure bringing some of the blood back in, though it's not as great. So a net pressure of about 10 milligrams of blood is 10 milligrams of mercury of blood is pushed out. So some blood and some liquid is pushed out on the atrial end. As it goes further down to the venous end or the vein end, the hydrostatic pressure kind of goes down because it's getting further and further away from the heart. So that hydrostatic pressure decreases, but the osmotic pressure stays the same, and that brings in more of the blood. And the main cause of that is these plasma proteins I mentioned before called albumin, or big Al as I like to call them. Albumin, through osmotic pressure, because more solutes in the blood than the interstitial fluid, brings in blood vessels and liquid back into the blood, causing the blood to be like that. This is very, very important with lymph tissue and lymph fluid, because not all blood or all blood fluid is brought back into capillaries. A lot of it, like 10% of it, is pushed into lymph fluids. We'll talk about more next time. And here's my references, and hopefully that was helpful. Thank you very much. Have a great day.